brief land acknowledgement that we read before every program here. The Berkeley Public Library buildings are built on the territory of Huchun, within the homeland of the Chochenyo-speaking Ohlone people. We acknowledge and honor the Ohlone people who have lived in Berkeley and throughout the East Bay since time immemorial. The library recognizes that Berkeley residents have and continue to benefit from the use and occupation of unceded stolen land, and we pledge to recognize the permanence and the rich contributions that the Ohlone people continue to bring to the city of Berkeley and throughout the East Bay. We celebrate the strength of the Ohlone people and their culture. The Berkeley Public Library is committed to helping create a more just and anti-racist society and to furthering the visibility and celebration of Ohlone authors who are specific to the East Bay and Ohlone culture in both our collections and our programming.
That's funny you should say that. I was just walking by that development complex by the water down there, where the sewers dump out, and they got all these tractors and concrete rebar going into building a Ohlone garden.
It's a little just conduits, yeah. So welcome to Talking to Science Bubble. This is a monthly seminar series that aims to share new research findings from grad students and postdocs at UC Berkeley with the general public and create constructive discussion about a variety of science topics. We have 2 speakers at each seminar who talk about their current research or a topic they find interesting, and it's organized by graduate students at UC Berkeley. These lectures happen on the 3rd Tuesday of every month at 5:30 here at the Central Branch. These programs are live streamed on the Science Bubble YouTube page, so you can view them later and share with others.
Oh, nice.
And we also have the listserv email served so you can join and keep up to date on the topics.
What was the YouTube channel?
The Science Bubble, right?
Yeah, we're going to have a link and everything at the very end that we're going to put on the screen.
All right, I'll hand it over to.
Thank you. And thank you all for being here. Like it was just said, we host a talk every 3rd Tuesday of every month. So if you're interested in hearing more about awesome science that's going on at Cal, both by graduate students and postdocs, please join the listserv that we'll have at the end, or you can Google Popping the Science Bubble, it will show up. And you can also visit our YouTube channel for an archive of amazing talks from the previous month. So for tonight, we're excited to have Lorenzo and Justin here with us to present.
Yay.
This is an open format talk, so we encourage questions throughout the presentation, both for our in-person folks as well as folks joining on Zoom. You can submit your questions either through the chat function, the Q&A function, or raise your hand if you're in person. And we'll also have time after each talk for more questions as well. So to start us off, our tonight's first speaker is Justin Willems. Justin is the PhD candidate in molecular and cellular biology at UC Berkeley, an Atlanta native. He received his BS in biochemistry from Georgia Tech. While in college, he worked both in a biochemistry lab on protein engineering and at Abbott Laboratories on heart pacemakers. He moved to Berkeley in 2018 and joined the lab of Wendy Shackman. He studies how cells in our body communicate with each other and how cells repair themselves when damaged. In a little time he gets outside of lab, he teaches at San Quentin State Prison, plays soccer with his friends, and spends time in nature with his dog, Calvin. So please take it away.
Are you familiar with connective tissue disorders at all?
I'm not, no.
Would you also make sure to share it to your screen to Zoom?
Oh, yeah, they did say we disabled when I tried to put
We'll quickly fix that.
it.
Hopefully you should be able to do it now.
Okay, let's try.
Yeah, we can keep our center view on the Zoom. You want to
Uh-huh.
share the desktop instead of.
To share the desktop instead. Okay. All right. Is that what you did? Cool. Okay, cool. My name is Justin. Yeah, my name is Justin. I'm a graduate student at UC Berkeley. Yeah, if you have any questions, you can just stop me anywhere in the presentation. But I'm here to talk a little bit about my thesis work on how cells repair themselves when they get damaged, and kind of how that process works. Okay, so I'm a cell biologist at Berkeley, so I think about cells. This is a diagram of a cell, specifically an animal cell. And it has two features I'd like to just talk to you about. It has an interior, and the interior has a lot of stuff inside of it, all these things that make the cell work. But one thing I'd like to highlight is that it has a lot of proteins. These proteins are like the nanomachines that make cellular processes happen. Like they're the things that make a cell move, the cell divide. All of these things are are orchestrated by these nanomachines called proteins. And then they also have like a boundary. This boundary we call the membrane. You can think of it kind of like your skin, and it keeps, just like your skin keeps your organs inside of you, this membrane keeps the proteins and all of the stuff inside the cell. Okay, but this is just one type of cell. This is, as I said, this is like an animal cell or your type of cell. Other cells can be a little bit different. So Lorenzo is going to talk about plant cells and probably microbial cells as well. Most of the rest of life is a little bit different in one key way. They have something called a cell wall on the outside of their cells. I think of this kind of like a suit of armor that goes on top of your skin like that. They keep those cells rigid, and they keep them more less flexible than say our cells. And they kind of change the way, this lack of a cell wall has changed the way that our cells move, the way they do things. So this, for example, is a classic video of a neutrophil, which is like this immune defender. There are these cells that patrol your body, and they look for invaders like bacteria. And I want you to watch the way that this cell, this neutrophil, moves inside of you. It has this flexible, because it has this flexible cell wall, it can kind of crawl. It can touch the red blood cells around it, and it can sense, oh, are these invaders or are these friendly cells? It can squeeze through narrow passageways. So in this case, this is the neutrophil cell. These are red blood cells. And then this is a bacteria right here that it's chasing after. So yeah, watch the way that it like kind of crawls along the surface. It's chasing after that bacteria. It's touching red blood cells, and it's like saying, oh, these are friends. And then finally it swallows that bacteria whole. Now try and imagine that process if it had like a suit of armor, like a rigid cell wall around it. Like so many things that you just saw there would not be able to happen, probably. So I kind of think of our cells like an octopus. This is a video of an octopus going through a maze. And you see the way it has these tendrils that like, it can squeeze through passageways. It can reach out and touch things and pull things back. Well, I think of the rest of life like these oysters or these clams, kind of, they sit in this rigid shell that keeps them kind of well-rounded and well in this rigid conformation, but they can't do these crazy movements that our cells can do.
Ooh.
So yeah, so I guess if you remember one thing from my talk, it's like we're made of trillions of octopi, little tiny octopi. The trees out there are made of trillions of tiny clams. So okay, but I just told you there's all these great things we get. Like in biology, you almost never get something for nothing. Like there's always a downside. Like what if you were going to guess, like what might be something negative to not having this rigid coating.
When they attack your own body,
They attack your body.
when they break out of your bones in mass in order to defend against.
Yeah, okay, so I guess that could be something like you can kind of get, maybe in certain conditions, some sort of autoreactivity. Is that what you're saying? Something like that? Or what do you,
Well, the COVID was the
Uh-huh.
serious cases were an overreaction.
I see,
That's why people's lungs shut down.
sure.
So one more time, what was the question?
Like, what is the downside not, what could you imagine is like a downside of not having this rigid shell? Or like, why would the rest of life, they have these rigid coatings? Like, what might that get you? Like, I just talked about all these advantages to being able, yourselves to being able to move like an octopi. Like, what are some downsides? Like, you don't ever get something for nothing, so like.
I mean, one, the thing that's interesting about the statement is, the question is that, like trees, we have bone cells, but I assume maybe the question is that it'd be more like a tree or fibrous.
Yeah, so that is like one of the, yeah, we wouldn't be able to move in the same way for sure.
Yeah, I think so.
Yeah, I guess one of the things I was trying to get at is that like these cell walls are kind of like a suit of armor. Like our cells are, if we don't, since our cells don't have those, we're like susceptible to damage from like our cells can break open much more easily. So that's one thing I was.
Bingo. I think that's what I was saying in a way.
Yeah, I think you were. I just
Yeah, that
didn't
was
quite-
one aspect of that, definitely.
Okay.
Yeah, so yeah, I mean, we have skin and stuff on the very outside that actually protects us. But inside, our cells are susceptible to damage. So if you just exercise for a few minutes and you just move your muscle cells, they'll break open. So this image I'm showing you is from a mouse that was exercised for a few minutes on a treadmill. And this is a picture of its muscle fiber. And what I hope you can see is that there's a huge gash in this muscle fiber after it's been exercised for a few minutes.
How big are the cells though?
These cells, these are really big cells. So I mean, like one of these nuclei is like 5 or 10 micrometers. So
Okay, that's a nuclei.
yeah, and so this whole cell, I mean, this whole cell is, yeah, it's a very, very big cell, but still probably not visible to the eye quite. So this is like magnified a lot. Yeah, so this gash is probably like, but I mean, it's maybe like a third of the way through the whole cell, right? Like it's a huge gash, but these cells, I mean, these gashes and these tears happen all the time. And I'd like to emphasize, this is not like an injury that doesn't get healed.
Right.
This is just normal. And within 24 or 48 hours, this gash will be repaired and sealed, and your muscle cell will actually even grow back stronger than it was before. This is like one of the leading theories for why you get that long-acting soreness after you like work out, is because you get these little tiny tears in your musculature.
Not a single cell.
Yeah, it's a little complicated because muscle cells are actually a bunch of cells fused. They do this weird process where they fuse and they form these really long fibers that are like multiple cells fused together.
Okay.
So that's why the nuclei are all, you can't really see any. So yeah, that is one odd thing. These muscle cells are actually, I don't even know, maybe hundreds of cells. They all fuse together into one giant long fiber.
Do they share a common wall, cell wall?
Yes.
Membrane?
Yeah, they do. And this is the cell wall.
Is that a broken?
Or sorry, cell membrane.
Cell nucleus up top? The
No,
nucleus?
in this paper, they actually, this is not, we're not gonna be talking about this today, but the nuclei will actually move towards the tear and like,
Okay.
so to help in the repair process. So
That's what I see.
yeah, so they get really close.
We do that in plants too.
They do?
Yeah.
Oh, that's interesting. So, so do plants also get tears?
Not
Yeah.
the same way.
Not in the same way, I see. So another instance where you get tears in your cells is during infection. So a lot of infecting cells, or a lot of infecting organisms like bacteria, they'll perforate your cells, and we don't really understand why they do it, but one theory is that they do it so they can suck nutrients out of our cells. So this is the cartoon of a person with pneumonia, and someone with pneumonia, their lungs fill with these bacteria, and these bacteria secrete these toxins that punch holes in our cell, kind of like a hole punch. They just punch holes throughout our cells. This is a picture of a membrane combined with this toxin, and I hope you can see it forms these rings that it punches these holes in our cells so that stuff will flow out. And one of the theories is that they're eating the interior of our cells, and that's how they incorporate our cells. But one thing that our cells have evolved is we've evolved repair pathways. So we're not totally defenseless. And there are these different repair pathways that allow cells to heal themselves when they get these tears or these hole punches inside of them. So here's a cartoon of a cell. It has a little tear on its exterior. And then this repair pathway occurs, and it's occurring primarily by these protein nanomachines that I talked about. These are the things that drive changes in your cell. They take processes, they drive processes in the cell. One of the proteins classes that I want to highlight are called these annexin proteins. The name doesn't really mean much, but they're just a class of proteins that are responsible for a lot of these repair pathways. And we'll talk about them a little bit later, but they bring the cell back into a state where its outside is sealed, and the cell can now go on and do whatever cellular process or important process it was doing for the body.
Is annexin like a sociological definition of the cell?
It's a type of protein. So it's just a class of protein that are involved with the. Well, I'll show some images of them later. So.
Are they as funny as the cartoon?
Are they what?
As funny as the cartoon one? No, never mind. I was just riffing. I apologize.
So, okay, but just to talk about maybe kind of the broader importance of these repair pathways are important, and when your cells have mutations or when your cells aren't able to repair themselves in the right way, you get this set of diseases called muscular dystrophies. So I talked about earlier that muscle cells are some of the cells that get the most of these tears and these gashes inside of them. Because of that, if you have a mutation or you have some error in your repair proteins, a lot of times it will show up as a muscular dystrophy disease because those muscle cells are the ones that are breaking open the most often. So muscular dystrophy is characterized by like your muscles slowly deteriorate over time until you have a worse quality of life and often premature death associated with your muscles just slowly breaking down. Okay, so now I'm going to talk like a little bit about my thesis work where I wanted to understand how this repair process is happening exactly. And one of the great ways that we have to watch this repair process are with microscopes. So this is a fancy microscope called a super resolution microscope. And what it does is it just magnifies the cells a whole bunch so we can see them clearly and we can watch them as this repair pathway occurs. And this microscope, it has a special addition to it, which is it has a laser that it can shoot. We can shoot it really hard at the cell, so hard that it blasts a tiny little hole in the cell. So we can shoot a hole in the cell, blast open, and then watch the cell try and repair itself so we can try and understand how the cells repair themselves. And we're just using a standard cell line that was donated many, many years ago. And we just, it's a standard cell line, a cancer cell line, and we use that to try and understand how some of these repair pathways occur. Okay, so let's get into this microscopy experiment. And in this microscopy experiment, we're looking at two cells here, and we're staining them with a dye. I'll go into a little bit of detail later what that dye is doing, but I'll just orient you here. This is one cell here, and right next to it, there's a second cell. This is that inner part of the cell. It has like all the proteins. This is the inner part of the second cell. And then this area around here is the outside of the cell. And the way this dye works that we're staining them with, it stains the cells and it gets brighter when it gets into the inside of the cell. So you can imagine if you were going to create a little lesion or a hole in the cell, the dye will rush in, it will stain the cell and get brighter, but we can kind of watch and see what happens. So I'm going to go ahead and I'm going to shoot the laser right down here, and let's see what happens. So I shoot there, and you get this kind of repair patch that the membrane kind of condenses, and you get this repair patch that shows up right on the edge of the cell. And that repair patch, I'll go ahead and play that again. It occurs very, very quickly. Within, if you look at the time scale here, this is like in 30 seconds. So the cell has to like seal itself and patch off that hole really quickly, or the cell is just everything is gonna start to leach out. So you get that kind of repair patch, it forms, but then in the second phase, you can see it gets shed. So you get the repair patch forming, and then in the second stage, the whole repair patch sheds off of the outside of the cell. So all of that damaged membrane, all of that damaged stuff gets shed off so the cell can go ahead and keep living. So just to go back over, you get this repair patch, that dye rushes in, and it only stains that repair patch. That's because the cell has a way to kind of sequester and keep the lesion from, keep that dye from entering totally into the cell, because it patches really, really quickly. And then within a second span, you get shedding of that damaged membrane into the outside area of the cell. Yeah. And so then I talked a little bit about this annexin protein. That's the protein that actually does the repair stuff. And to orient you again, this is the cell. And then this is the outside of the cell. And I'm not going to go into how exactly we do this, but we have ways of specifically staining just that protein. And it's stained in red in this case. So we can watch and see what this repair protein does. So I'm going to shoot the laser at the bottom of the cell, and hopefully you can see that this repair protein like shoots towards the repair patch and does its activity, which I'll talk about what we think is happening. And then it is shed, the damaged membrane plus the repair protein is shed off.
I'm confused. How long does this process take?
It's 10 minutes.
10 minutes.
Yeah.
10 minutes. Okay.
Yeah, so it's pretty quick.
All right.
In your body, how often are you getting these tears?
It depends on the specific cell and the specific context. So people have done, they just take cells, say, from your muscle, and they, especially after you exercise them, about 20% of the fibers are permeable, that are basically ripped open at a given time. So quite a lot.
Wow.
In your heart, like say your aorta or something like that, it's less, but it's still 5 or 10 based on the permeability. It's about 10%. So it depends on the cells, but it's quite frequent in certain tissues.
There's like so many different heart tissues.
Yeah, and especially in the heart, because it's like driving blood like around that blood driving against the wall of your is thought to be able to kind of exert a force that shears those cells a little bit.
My theory in heart sciences was heart tissue type.
Mm-hmm.
And so, of course, there's some things that are genetic and some things that are not in many of the theories, but there's so many different types that it becomes hard to study cells in salt contexts and in especially vaccination process.
Mm-hmm.
So I assume maybe you had some pretty heavy experience with RSV suspects, maybe in the college system or in the university when working.
RSV, you mean the? I don't actually know what that means.
I guess the RSV vaccination.
Oh, like the virus.
There was like 7,000 vaccinations, and 100 of these vaccines would do heart and feet and skin, and they would target all of the spectrums of the body. So maybe you got to work with a lot more of this practice over.
I didn't know. I'm kind of a basic biologist, so I don't really do very much clinical or anything like that. Yeah, I don't really talk to humans that much.
Oh, hi. You're talking to humans for the first time in Helton.
I'm just an emoji character.
Yeah. So,
You're in the life science building, I'm guessing?
yeah.
The both of you, maybe?
No, actually, I'm in Mircobes. I'm in a national lab offshoot.
Cool.
So this process is happening constantly. right?
Yeah, exactly. Depending on where you are in the body, especially a lot. And
On
if
a
you're
much
working,
wider scale than I would have imagined.
yeah,
20%, you said?
yeah, in a muscle cell, that was done after, I think it was in a rat, and the rat would just ran for a few minutes, and then they cut open the fiber and looked. So it depends a little bit on how much that muscle has been working, but it's quite frequent, especially if you're like moving and exercising.
I'd like to see a whole gradient scale on that. now.
Do you know is the speed at which the repair is done is different if you're awake and active versus if you're sleeping?
I don't know about that. That's a good question. Yeah, there could be some relationship there. I would imagine that in general, just repair occurs a little bit less frequently when you're asleep, because a lot of the cell types that are involved with this, for example, your muscles or the cells that line your veins and your arteries, we call them endothelial cells. Those are like your blood is not pumping as fast, your muscles are not moving as fast. They're in a state probably where they're less likely to tear open in those kind of situations. Yeah. One other case where it's known to happen is just by cells migrating. So a lot of your cells are just migrating around, and those cells are also susceptible. So yeah, I'll just talk a little bit about what we think. I can't present all of the data, but just our overall model for what we think is happening when a cell is repairing itself. But I think it's kind of amazingly analogous to how a wound on our skin is healed. And I just using that as an analogy because I think it's something we've all experienced, where we have a cut on our skin, and then initially you'll have blood flowing out of the cut, right? That's normal, but then pretty quickly you get a scab that forms. And that scab is a block that prevents your blood from continuing to flow out, and then you don't bleed out. And then in a longer second phase, the scab is slowly broken down and it comes off of your skin as a scab. Now, on the level of an individual cell, we think like an analogous process happens where there's a break in the membrane, and these proteins, like these annexin proteins I just talked to you about, they quickly move towards the lesion site, the break in the membrane, and they form a patch, kind of like a scab. And actually, people in the literature also use the term scab interchangeably. So I think people are already thinking about this kind of analogously to repair on your skin. And in a second phase, that's just a temporary structure, just like the scab. So you need to get rid of all that damaged membrane, all that gunk, and it gets shed off as these little pieces of membrane flying off of the cell.
The annexin forming a patch, like actively or passively? Like I would think of blood as like it hits the air, it starts to clot. Is that kind of what's happening with the annexin, or is it more of like an active, like somehow they're detecting like there's pressure loss in the cell or something, and they go and try to find the hole?
Yeah, there's something I haven't told you here, which all of this is based on a metal called calcium. So the calcium concentration is really, really high outside the cell, and it's low on the inside of the cell. So when you get a cut in the membrane, most of these repair proteins are sensing is a massive influx of calcium into the cell. And so that stimulates this whole kind of patch formation. Okay, so just thinking a little bit about like what we think some of the more useful applications of some of this kind of research would be. this was a study done by Elizabeth McNally. She's one of the leaders in the field. She's a clinical scientist at Northwestern, and she works on a lot of muscular dystrophies. And the experiment she did is she just took mice, and these are mice with muscular dystrophy, and she either injected them with a control or she injected them with these annexin proteins. And then she put them on a little tiny mouse treadmill, both of them, and allowed them to exercise. And what she found, what her group found, is that the mice that were injected with the annexin, their muscles healed a lot better, they restored a lot better. So it seems like even just giving, and hope maybe theoretically this might work on a human, just giving people with these kind of diseases back some of these repair proteins might supplement and allow their cells to heal when they have these kind of defects.
They don't need to be gotten into the cell to do that. They can do it from the outside.
It's surprising. Yeah, you would think that they would have to be active right there but apparently they'll work and they'll find their way and they'll do their thing. So I'm kind of surprised by that as
They'll
well.
find that calcium coming out and
Yeah,
wait a minute, I'm confused.
because you would think that so what I told you, right, is that they're triggered by calcium, right?
Right.
And so they're injected into an environment with really high calcium. You would think they would activate just right there as soon as they enter. But I mean, I guess somehow, she injected them intravenously. This wasn't even into the muscle, and they seem to find their way and make a substantial
The straight
difference.
annexin with no kind of
carrier or
penetration.
nothing. Yeah,
Cool.
it's kind of surprising, but yeah.
You guys think then that when annexin gets released from the cell during shedding, that it just kind of makes its way into another cell again, or that it just gets degraded in the ether that is the extracellular space?
We think it gets degraded. Yeah, it's kind of like apoptosis, any of these processes where there's this kind of shedding, where there are these dedicated cells that just eat huge amounts of stuff and just chew them up and degrade them. Similar, there's these cell death processes. I think it's about 1 or 2 kilograms of your cells die every day. And those have to be degraded in an efficient way. And you have these cells who their job is just to eat these dead cells and this kind of debris that flies off of cells and degrade it. So I think that's what's happening, yeah. We actually isolated some of these particles that come off. Well, some of these particles, and they have these signals on the outside. They're called in the field eat me signals, and they're signals that tell other cells to eat them up and degrade them. So yeah, presumably they're getting degraded. Okay, so that's all I have. I work in this great lab, and there are all these other people who I talk to all the time and give me ideas. And we have collaborators from different parts of the country. Chris is the one who taught me that method that I showed you, that laser ablation method. He's at Emory, and also we have collaborators in Maine. So yeah, and I'd like to thank our lab manager as well. Yeah, so are there any questions? Thank you.
What's your thesis?
What's my thesis?
Yeah.
It's on the shedding, the specific shedding process that occurs. How does that shedding process occur during repair? So how are these?
The mechanisms of what you just showed.
Exactly,
is that
mechanistically what's happening.
Yeah, very interesting.
There's a couple extra parts that I didn't talk about, but there's some other proteins besides the annexins that also join the annexins there and do some stuff to make everything work together and happen efficiently. Yeah.
First question is the eat me signal ubiquitin?
It is not. It is a lipid called phosphatidylserine, and that the phosphatidylserine, normally you have this lipid and it's on the inner leaflet, And then when there's this process activate, there's a scrambling of the lipids on the membrane. Yeah.
And the second was, do these, like when they form their, the plug and the patching and such like that, are they like dimerizing? Are they like connecting to each other, or like what's the process?
Yeah, so so this protein is actually a dimer of dimers, and they bind to like the membrane, that outer part, in the presence of calcium. So what they do is they kind of form a cross link, and it's kind of like a big non-covalent, or it's just like a big cross-linking reaction that occurs that right there locally. If we can reconstitute it in vitro, where we just take isolate that membrane component and mix it with the purified protein right there, and when we do that, you form these patches. So you can see this patching effect that occurs just with lipids, or just with the membrane, and just with the annexin, not even inside the cell. Yeah.
Going off of this skin wound analogy, if for the cell membrane, you have injury and you have the plugging scab removal, and then if you happen to injure it at the same spot again, would it be faster to patch it, or would it, trigger something else, or just like a bad cell, we need to get rid of it?
They don't fully understand how this works, but definitely muscle cells, that's true. So if you have a lesion in a muscle cell, I'm pretty sure I remember this right. Yeah, you're less likely to get those tears, and that's part of like strength training and things like that. It's like you're less likely to get those tears in the muscle, and I don't think they fully understand how that happens. It might be some of this repair process, but it might also be these proteins that kind of harden the muscle in a way, so you don't even get the tear in the first place. Yeah.
Genetics are ridiculous. It's a really hard study. How each piece of energy is grown, essentially. Grown.
Yeah.
And creating a human being from a insemination process to create a embryonic phase that writes everything that the human body is going to do, almost. Supercharging.
Yeah.
Supercharging everything with the memory card.
Yeah.
Again, with the connective tissues, I would suggest just taking a brief look in that side alley on your research, at a cursory glance at what connective tissue disorders are. You might find that really interesting.
Yeah. There's a whole component to like how the cells, especially in muscle, like when they get damaged, their connections are what causes some of the most common to the connective tissue, or what cause some of the most common muscular dystrophies. There's this muscular dystrophy called Duchenne's muscular dystrophy, which is like probably the most common. And it's the way it starts is just this: the muscle cell is not fully hardened, I guess, or like it doesn't connect properly, so it's more likely to tear in the first place. So definitely how the cells connect to the connective tissue is very important.
If there are no more questions, then I think let's move on to our next. Give one more round of applause, and then we'll move on to our next speaker.
Thank you.
Thank you.
Definitely.
All right, I'm going to start introducing Lorenzo while I go ahead and get set up here. So our next speaker is Lorenzo Washington. And Lorenzo Washington grew up in a small town outside of San Antonio, Texas. Lorenzo has never been far from gardening, farming, and playing around with whatever he could find outside. Over the years, his curiosity for the outdoors and proximity to agriculture led him to study both environmental and animal sciences at Texas A&M University. There, he was exposed to research that inspired him to continue on to doctoral studies at the Department of Plant and Microbial Biology here at UC Berkeley, where he focuses on genetic and biochemical aspects that influence how plants navigate beneficial relationships with microbes in their root systems. As an avid practitioner of judo and lover of live music, he has found many ways to recharge from his research work. And true to the Bay Area, Lorenzo also enjoys a variety of outdoor activities such as hiking, biking, and mushroom foraging, and finding whatever excuse possible to make a trip to the sea eras.
All right, take it away, Lorenzo.
Nice friends.
All right, next. So thanks for coming. I appreciate y'all here to listen to me talk a little bit about some research that's pretty adjacent to a lot of the work I've done over the last several years. Just to start off, if you've ever gone through the woods on a nice hike or walk before, you're probably familiar with just how quiet and serene it can be. Maybe you hear the breeze flowing through the trees, another hiking pair kind of talking a distance away, but it's pretty peaceful, right? Except for the plants and all the other organisms there, it's kind of like being in the middle of New York City. You're surrounded by all these other people, except unlike us, they can't move around near as much. So they're vying for sunlight and nutrients in a way that we aren't. And I was always wondering how these plants, how these organisms even manage all of this when they can't move, they can't speak. At least I used to think they were just kind of silently standing amongst these crowds. Until I started to look a little more closely underground. So plants might not be able to talk, but they can communicate and even negotiate when we kind of give them a more human lens at least. And they figured out how to navigate this diverse, chaotic world that is the soil ecosystem through their roots, which can be like a home to friends, can be a busy marketplace with shady back alleys, or even just a place microbes hang out for the time being. So how do they manage to sift through all of that noise and make sure who they want to stick around does and who they don't doesn't? Spoiler, the fundamentals aren't so different from how we form our relationships. So let's get into it. Thank you for the nice introduction. The kind of punny title of my talk today is Putting Down Roots, how plant roots form intimate relationships with microbes. So really quickly, why roots? Why are roots so important? They're actually really, really important for the soil ecosystem. So if you think about soil, it's almost like the ocean that's not the ocean, right? It spans a huge chunk of the world. It's very deep. It has a whole lot of living organisms inside of it, from very small all the way up. I think the kind of pop sci estimate is like 1 teaspoon of soil can have about 1 billion microbial cells, and the mycelium, so the fungal threads that run through there, can also equate to like kilometers nearly. So it's essentially every time you're walking, you're literally just walking over universes, microbes. But we're just so far in a scale above, we don't really connect with just how diverse and chaotic that environment is. Roots are really important for this because that that huge universe of all these living organisms is also like space where there's just a lot of empty stuff. If you're a bacteria, 1 centimeter is further than you'll move in maybe your lifetime, depending on how long you live, and double. And so just navigating a soil environment is really difficult. But if we have a root that is so conveniently placed through, it is almost like a city stretching across this desert. So roots not only influence soil structure, but they're important sources of nutrients for a lot of these organisms. They provide sugars, proteins, and a whole host of metabolites, so different chemicals they produce that we're just beginning to understand. And yeah, they are kind of these population hubs for microbes, both fungal and bacterial, which means there's probably also a lot of viruses, there's probably a lot of protists and nematodes. We just haven't fully understood the depth of these relationships quite yet. And they can also alter which of these microbes happen to be growing in the soil, not only through just providing these nice sugars, but they actually can, over time, filter for a specific kind of microbial community, which is pretty interesting. Oh, and one last thing is we don't really see them usually, so we don't get to appreciate just how much space roots can take up. For a lot of plants, at least the ones that, trees especially, and a lot of the ones that we don't grow for crops, their roots can be 2/3 or more of their actual total size. They can extend several meters down into the soil or out into the soil. And there's a whole lot of diversity there. It's just unfortunately, it's really hard to study because I don't know if we would like to try and dig up a couple hundred of those to do a study. There's a reason why it's a little behind in some other respects. So now those cities, right, there's a whole host of different kinds of microbes that might be living there. So who are there, right? Who are the plants conversing with on their roots? So we have three rough classes that just about any living thing can fit into. But in this case, I'm specifically talking about bacteria and fungi. And so the first class we have are pathogens. Probably familiar with that word. These are the bad guys, right? They cause disease. They poke holes in our cells and steal nutrients from them. They grow in places they shouldn't be growing. They start taking up nutrients that we might be sending, or plants might be sending in other respects. Or they could even be killing off a lot of the good guys. And lead to issues such as leaf spot, root rot, all sorts of a host of bad issues. There's also commensals, which is a fancy word for saying the okay guys. These have little to no effect on the plant, depending on the context. They might, but they themselves benefit from associating with it. They get a nice apartment in the city, so to speak. And I just kind of call these freeloaders, at least until they aren't. They're just chilling there, waiting for the opportunity to shine or peace out. And then the last one we have are beneficials, or the good guys. These benefit the plant in some way upon association, and we call these the good ones. So they could provide nutrients, they could provide defense against other pathogens, or even provide resistance to certain stresses like drought or heavy metal in the soil.
Why was there a canopen soybean nodule in there?
Oh, yes, I will talk about that a bit later. So, but brief preview, nodules are one of the particularly intimate forms of relationships plants form with bacteria. And nodules are kind of like the really prime time apartments in the city. They're special structures that specific kinds of bacteria can live inside. And they actually make nutrients for like specific like nitrogen fertilizer type nutrients for the plant in exchange for sugars that I'll talk more about in a bit later. So why do plants even go through all this trouble, right? One thing is, unlike animal cells and animals in general, plants don't really move. So they have to be creative about solving stresses like not having enough water, pathogens showing up, not having enough nutrients, right? And the microbial communities in and on plant roots, as well as their leaves, are essential for robust health and development. So in that respect, they're actually not all that different from us, right? We have all sorts of microbial communities living in and on us. There's bacteria in our hair, armpits, stomach, right? Helping us digest food, keeping pathogens from growing on us. And plants are very similar in this respect. The difference being, as opposed to us, is how they get their nutrients and their general lifestyle, which influences how they navigate these relationships a little bit differently than we might. So how do they speak and navigate all these relationships, right? We know plants don't really make noise, at least not that we can hear. There's some cool studies that show they do make noises, but it's probably just like passive processes. Unfortunately, or fortunately, depending on what you liked in school, it's biochemistry. I wasn't a big biochemist, but you gotta learn at some point, right? So turns out there are about as many words as there are molecules, and then some. So plants and microbes in turn can send out all sorts of different kinds of chemical molecules, like auxin, for example, which is a very classic plant hormone. And for the plant, normally this used to just regulate how they grow their roots and which direction they grow above ground. Also, however, there are quite a lot of bacteria that have learned how to make auxin or similar kinds of auxin, or even eat it from the plant, which means they now have a certain kind of relationship with the plant that can be good or bad depending on certain contexts. Additionally, things like the really long word over there, lipo, keto, oligosaccharides, which basically just means they're little fatty sugars that happen to have a little bit of chitin in them. Those actually are fungal signals. Fungi have been using those signals for a very long time, and plants at some point happened to also use them. And then some bacteria figured out how to use them. And now they all kind of can talk or at least understand each other in some respect together because they share this kind of biochemical language. And also, let's see, missing anything? Yeah, and so much like our language, these words can be, they're very informative. They can quite literally, in many cases, cause harm to either party involved or allow them to safely come closer together. And I'm just going to keep saying speak and talk for the rest of this presentation instead of like they produce toxin, jasmonic acid, so it's way easier. Much like our conversations, their conversations kind of follow the same basic approach. So someone approaches you in your proximity, and one of the first things, unconsciously or consciously, you might be doing is, oh, hey, are they being aggressive? Or has that person punched you in the face in the past? And if the answer is yes, you're going to respond accordingly. If you're a human, you might run or fight, depending on how you feel. If you're a plant, you can't really run, so you just kind of fight. And they have a whole suite of processes that they can do to fight off different pathogens, from literally just spraying hydrogen peroxide to destroy their own cells, to putting up more defenses, strengthening their cell walls, whole host of processes. Now, in the event that this person, this bacteria, for example, hasn't been aggressive in the past, or if they don't seem to be aggressive right now, okay, let's hear them out. Let's talk a little bit, right? Let's exchange some molecules, see what's going on. Maybe they're a little bit boring. Okay, cool. Nice talking to you. See you next week, right? See you on the wayside. Or maybe this conversation is actually interesting. There's a spark. You sense something that could be nice. You want to go continue on this talk at a bar somewhere else, right? And so the next thing you ask is, do they fulfill some of my needs? In the case of the plant, are they providing nutrients I need? Are they really good at competing against something that constantly likes to hurt me? Or maybe it's just really good given the soil chemistry I'm in. If you're someone like us, maybe it's good companionship. Nice buddy to talk about. They really enjoy the random thing you like to talk about all the time, right? And if you notice with the little arrows, this, as well as other parts of this process, is a constantly occurring thing. There's constant check-ins involved, just like our relationships, where you kind of have to keep talking to each other. Hey, how's it going? Hey, do you have other needs that I need met? Do you want to spend more time together? Do you want to go get a drink? Did you like the pizza I bought you? Those kinds of questions. Plants and bacteria and fungi have similar kinds of conversations through their biochemistry to kind of make sure things are up to snuff and these relationships are going according to plan. Yeah.
What is the timescale?
Remarkably fast sometimes, not so fast other times. So in a little bit, I'll talk about a couple of relationships. One of which that happens on the scale of weeks, one of which can happen on the scale of days, which for plants is extremely fast. And one of my favorite facts is plants can, similar to how they were saying earlier, where nuclei can move accordingly. In certain situations, with certain kinds of relationships, plants can navigate their nuclei to where these microbes are, and they can kind of decode calcium signal spikes that translate into other responses in minutes to hours, which is incredibly fast for plants. So there's a huge range of timelines that these relationships can be happening.
Wow.
Yeah. And another way that plants also kind of offer up these conversations is just they're a place full of food for a lot of these microbes. Or if you're a microbe who eats microbes, full of other microbes to eat. So all of those molecules I talked about, not only can they be words in a language, they can also be a source of nutrients for a lot of these creatures. Or just straight up sugars that the plant is either sort of consciously pumping out into the soil around it, or just cells that have sloughed off as the roots have grown through the soil. So these kind of roots are kind of cities in their own right, in that, there's a whole lot of food there's a whole lot of conversation happening, there's a whole lot of people, and it's all generally a mixed bag, right? Now, I'm going to zoom in a little bit on two particular relationships that plants have formed, which is also what I've studied quite a lot during my PhD. And these relationships are very intimate insofar as you'll see in the next slide, and you can see in some of these images, plants actually allow these microbes to live inside of them, which they only do for a very, very small subset of that city that lives on their roots. Most microbes are on the outside. Very few get privileged access to enter the plant kingdom, so to speak. And these two are truly longtime friends in that the fungal relationships, the one you can see up in the top right, all the green is a fungus. You can see how it eliminates the root cells, or this image here. That relationship is about 450 million years old. So older than roots even, which is pretty cool. There's fossil evidence to back that up. And the fungal, or this fungal relationship, ones you might see in the forest, where chanterelles and morels are growing off of plant roots, or the bacterial relationship that I mentioned with the nodule, so legumes, peanuts, and peas form these relationships. That's about 50 million years old. Young by the other standard, but still pretty seasoned in terms of relationships.
So the nitrogen fixing nodules common amongst the legumes is all due to bacteria.
Yes, actually.
That's what you've been saying this whole time. Cool.
Yeah, so we say nitrogen fixing plants, but really they're just managers. And they're hiring bacteria to do nitrogen fixing for them.
And that's all down in the roots.
All down in the roots, yes.
Okay.
Although there's a little bit of leeway there, in that there's also some bacteria that will fix nitrogen not in those environments. It's just much smaller amounts. And they can even grow on things like leaves or just on the surface of the root, but it's just much less that they're doing. But they do it through a similar process. The reason that these plants form nodules specifically is because the chemical reaction to make that nitrogen that they're giving to the plant is literally taking the nitrogen in the air and breaking the triple bond and turning it into a form of nitrogen that the plant can actually eat. So it's kind of magic in a way.
Interesting. I didn't know that.
But that's really hard to do in an oxygen-rich environment, which is why they get these special compartments that when you cut them open look a little bloody, because they have a version of hemoglobin, which slows down the diffusion of oxygen. So these plants are real crafty. And they solved a pretty interesting problem in a very interesting way. And there's legumes are kind of the only, there's like one other exception branch of plants that have actually evolved that relationship. Funny enough, a lot of the genetics were borrowed, so to speak, from the fungal relationship. They kind of duplicated and over time kind of gained this new function to allow this relationship to happen. So these relationships are actually connected themselves.
What was the type of fossil?
I forget the exact name, but essentially there's this fossil from like 400-ish, 500 million years ago. And this is when the first land plants were forming before they had roots. They just had like rhizoids. And in this fossil, in this plant cell, they see basically what you see in the top right almost. They see these like tree-like structures of a fungal cells growing in a plant cell in a way that doesn't look like an infection and is remarkably similar to what we see with this relationship nowadays.
Like amber or like remineralization?
I think it was an amber fossil.
Yeah, I can imagine you'll see that.
Right. So these two kinds of relationships, a really mouthful of a word, mycorrhizae, which is literally Latin for fungus root. So you can call them fungal roots if you want. And rhizobia. I don't know why they got that word, honestly. These are the two kinds of associations that plants have a particular kind of interest in, or at least some do in the case of the rhizobia. Mycorrhizae are a fungal association. I like to describe them as kind of caravan traders. They show up, they extend root systems over distances that they normally wouldn't be able to cross. It can exchange things like nutrients and phosphorus and nitrogen, even between plants or from different sources that the plant normally wouldn't be able to access. They can also aid in water uptake. So they're kind of like the travelers from a faraway land showing up to give their gifts in exchange, of course, for sugars and a nice place to live. Rhizobia, the bacterial association, the ones who fix nitrogen from the air and turn it into fertilizer, they're a bit like live-in chefs or alchemists. They show up, they ask for a place to live and do their work, and some sugar to keep them going, and the plant obliges. Now, if you'll notice, these relationships share this similarity in that they're really letting these microbes have premier access to their cells, right? They're growing, in the case of the fungal relationship, all in between the plant cells themselves, and to form their trading structures, they actually penetrate the cells to grow this temporary structure to exchange. And the other kind of relationship with this, they surround the cells to do this, but they still have some penetration as well. And in the bacterial case, they're actually forming these threads that go through or kind of groups that penetrate into the root before forming these kind of nodules, which are localized factories for nitrogen production. So, considering plants don't want to let in not friendly microbes that are not giving them all these important nutrients, right? This is kind of a risky business. You can't move around. You're literally just kind of opening the doors to your otherwise well-defended kingdom, and to at the chance that you might be letting in something nice, right? So how do they actually make sure this doesn't go wrong? Well, it all comes down to this conversation, right? So those molecules I talked about earlier, there's a very extensive signaling process that happens that can even diffuse out into the soil. And these microbes and the plant in turn are very sensitive to this. So the microbes can sense the molecules the plant is sending out, and the plant, they send molecules back to the plant in turn, which tells the plant, hey, I'm a nice guy, let me in, right? The plant then lets them in a little closer, they exchange more signals, and kind of verify the initial call that these are good guys. And then once they do start to form this relationship, they respect each other's boundaries. So these kind of growing in between the cells and this kind of thread that grows into the cells is actually very heavily monitored. It's very well regulated. It's a barrier that plants do not like to break. So technically speaking, they're never actually in the plant. They're just in a very complex tube that the plant let them be inside of the plant. It's a little weird like that. And these have to respect those boundaries, because if they, as soon as they break those barriers, the immune system activates and the plants want to get rid of them. They'll either blast their cells, start all sorts of other processes they would do in the case of actual pathogen. And also, much like the case of like a live-in chef or a caravan trader, they need to prove they can be counted on. So these relationships are monitored in the way that plants can kind of over time understand how much fertilizer, how much nutrients they might be getting from these relationships, like these individual factories or these areas where they're forming relationships with the fungi, and in turn, how much sugar and fat they're sending for those resources. And they can kind of make these assumptions based on other things like, hey, I'm kind of feeling really sick, or I'm not making a lot of photosynthate anymore. The sun's not really shining too much. I don't know if I can keep paying these guys to make nitrogen in my roots. Maybe I got to kick them out, right? Or in the case of maybe an actual pathogen showing up, they just kind of shut up all their walls and want to reserve the resources. So they might actually stop some of these associations. There's to kind of humanize them a bit, they can actually kind of choose when to let these relationships progress or not. We still don't fully understand that process, but it's very clear that there is a sort of extensive monitoring and kind of decision-making process going on. This slide's going to be a bit messy, but I like this because it kind of summarizes all of that in a nice analogy. So you can imagine the wall here is the wall into the plant kingdom, this wonderful realm on the other side. And outside of the wall, much like you see in many RPGs or medieval movies, there's a whole host of people living in a town. And this is the microbial city. It's full of all sorts of random people. There's also some not so nice people looking to break into the plant kingdom and steal its resources. There's some caravan traders who are showing up from the faraway land. There's some chefs and alchemists showing up to offer their services to the plant kingdom in exchange for entry, right? And so they let that be known. They send out those signals to say, hey, we're good guys, let us in, open the door. I promise we'll make it worth your while. And the plant kingdom has a whole system for sending that message down to the king, or in this case, the nucleus, and asking if it's okay and can we actually do this. Messages come back and we go, yeah, sure, open up, let them on in, but be careful. We can't just let everybody in through the door. So make sure it's just the traders and the chefs, right? However, the plant kingdom is also unabashedly a surveillance state. It knows where you're at, it knows when you're there, it knows what it's giving you, and it will kick you out if you cross any boundaries, or unless you're really good at it. And so that kind of summarizes that whole conversation process happening. There's a lot of this back and forth going on. And this is to kind of convey the scale of this. You can imagine this is just like 3 or 4 cells. This is happening across an entire root system. And like that first image I showed you, root systems can be, like some grasses have roots that can extend 5-10 meters down into the soil. There are trees, of course, which have extremely extensive root systems. Oftentimes roots can actually live in much longer than above ground tissues, right? There's perennial plants where their above ground tissues die off and their root systems live on. And so this is happening at essentially every single cell across the entire root system nearly at any given time. So the plant is navigating a whole host of conversations at once and decoding all the biochemistry and the genetics, and do I have enough nutrients? Are these the right guys I want to talk to? I don't know how I'm feeling today. All of that is happening all at the same time to decide who it wants to keep around and who it doesn't. So yeah, plants are handling a whole lot of information and navigating a whole lot of relationships that we don't give them credit for.
I noticed you don't have viruses in there. You got fungus and bacteria. Are the viruses never useful?
Oh, no. There's extremely complicated relationships with viruses. They're just unfortunately really hard to study because oftentimes viruses are obligate, so they'll only live like inside of certain bacteria or fungus, which makes them really hard to study. But there are situations where like there's studies that have shown that certain bacteriophages, so viruses which feed on bacteria, are contribute to enabling plants to resist certain kinds of diseases.
Do they borrow it from what they eat?
No, and just like their presence in the soil will actually lessen how much of the bad guys.
Gotcha. It's a difference
around.
Against the.
Yeah.
They battle each other and then they lose cesareans. They go away.
Pretty much. It's and so the plants aren't necessarily actively directing the viruses, at least not that we can understand, but they could be doing things like attracting bacteria that would be hosting those kinds of viruses, right? Or attracting fungus that could be hosting the kinds of viruses that feed on bacteria and vice versa. These are possibilities, but these are also really hard to prove experimentally. So
I didn't even know there were fungal bacteria. Thank you.
Yep. And kind of before I wrap up in the summary, how do we even know these things and why are they important? As far as how we know these things, we can think largely molecular biology and microbiology, which is a combination of understanding genetics, so understanding the DNA, RNA, so the blueprint, so to speak, that we have in our body, what it's doing at a given time, what that means for the proteins, so all those little machines that are doing things in the plant cells or the microbial cells, what they're doing, and the function that those have. And so we can do those in a lot of ways. We can grow really fancy systems to monitor processes as they happen in real time. We can use microscopy, staining to see where these relationships are in the root, or how these alter cells within the plant, or even just looking at soil samples and seeing what's in the soil. And we also use, especially nowadays, a lot of genetics. So there's a technology called sequencing, which lets you kind of read all of the DNA in an environment or in a sample that you send it. So say a root sample or a soil sample, and the computer can help you parse out all of those reads of the DNA and tell you what they might have come from. So they go, hey, this came from a sunflower plant. This came from this kind of bacteria, or that kind of bacteria. It's a rough idea. It's, I think another scientist used a great example of, it lets you differentiate like carnivores from herbivores, but you can't differentiate like a bear from a dog, for example. So you get a decent idea of what's there, which, considering just the sheer amount of microbial life that's in the soil, is pretty good. And we can combine all those technologies with experiments in the lab, experiments in the greenhouse, and experiments in the field, or just kind of ecological studies, to combine all this information and start to try to understand what exactly these plants and these microbes are going through, how they're navigating these relationships, how they change according to nutrient availability, how much water is in the environment, what the soil is like, what happened to be growing there the year before, right? All these questions. And a great example of how why these are important, aside from these relationships often contribute positively or negatively to how much food we can grow each year, which is pretty directly important to us, not to mention supporting the ecosystem and environment that we all go through, especially here in the Bay all the time if you like to hike. But as I mentioned before, and one of my good friends who recently started on this project, Dr. Sierra Ragland, has been working on, is this larger One Health project, which acknowledges that through these microbial communities, there's a lot of connection between us and the environment, and as well as animals, other animals that live in that environment, through the microbes that are present there, right? Microbes in the soil can influence what kind of nutrients plants are able to take up in terms of like mineral capacity, so zinc, magnesium, calcium, these things that change the kind of quality of food that we eat, right? Also the different kinds of microbes that are on the food, or in the food, even through, even when eating it, can it change what kind of stuff is growing inside of you, inside your gut tract. Same thing can happen for other animals, can change their microbiome. So there's a lot of connection that we don't really understand. And work like this is trying to start to unravel that really complex network. And just to summarize really quickly, plants can decipher precise communications at small scales along the roots, and also have ways to filter these microbiomes, whether through conversation or offering certain kinds of food up, so to speak. Many of these relationships can differ across contexts and have space for a wide range of behaviors. Plants are generally talking to people they want to be nice to them, but there's also not so nice people. There's microbes that are nice in one context and not nice in the other, or nice to one plant and not nice to the other. There are plants that want something in the dry season versus in the wet season, right? There's a lot of complexity and contextual relevance to how these relationships happen. And additionally, as we've gotten more ability to kind of understand the universe that is the soil ecosystem and the microbial ecosystems that live on and inside of us, as well as plants and animals, we are starting to understand more and more that the environment is this very interconnected network of dynamic relationships, not just kind of nutrients passing from one source to another, but maybe a bit more of an active process in some respects that can kind of ripple out and have effects that we otherwise wouldn't have been aware. And yeah, that's the end of the talk. A lot of the work I've done over the years is not just by me. I've had a lot of help from some wonderful undergraduate researchers as well as other doctoral researchers. And aside from the plug for giving SHUNY to the Agrarian Land Trust, these are all the people that give me money, and do this kind of work.
Yeah.
And with that, yeah, any questions?
But like lookalikes, so you know, if you have a fungus or a bacteria that's like, oh, I'm gonna act like this thing you like and sneak in and steal stuff. I guess like, how common is that, and like what do plants do to be able to say like, oh wait, you're not who you said you were, and like without just kicking out everybody?
Yeah, see, that's a great question. I think so. There are cases of like lookalikes. For example, the nodulation relationship. It's not just like one bacteria that does this. There's an entire kind of host of bacteria that can form these relationships, and that differs based on the plant. So different plants will form relationships with different bacteria, and even within that, certain ones will be better at providing nitrogen or might do it cheaper than other ones. And there are some that are really good at like being like, hey, I'm here to fix nitrogen. Not really. I'm not going to do a great job at it. And so there's a lot of like kind of situations like that happening. And there are microbes that will,
But those would be truly predators.
I guess, closer to parasites, because they're not actively hurting the plant. They're not like damaging the plant to get these nutrients. They're just tricking the plant into giving them the nutrients, if that makes sense.
Yeah, it's working cooperatively in a way. Yeah, not a problem.
And then there are also some kinds of microbes that can produce some of these molecules that like bees would use, for example, to communicate with the plant to try and lower some of its defenses. But plants have a very complex recognition network to kind of decode these things. So not only are they being like, oh, you're getting this molecule, but they're also like, hey, do I sense chitin? Do I sense bacterial flagellum? Is this part of my immune pathway reactive to it? Is that part of my immune pathway reactive to it? Is it reactive to this very specific random interaction I have in my immune system? No? Cool. All right. And so there's like, it's really complicated that we're starting to understand just decoding of all these signals that kind of limits, at least just kind of opening the gate for certain pathogens that might be wanting to just fake signal with it, I suppose.
So I'm imagining there's like a plant and it like wants some bacteria to fix nitrogen that sends out some signal or something like that. You mentioned that like bacteria can only move like very small distance. Like how do these bacteria or these fungus like even get to the right place so they can like be there or something? Does that make sense?
Yeah, no, that makes a lot of sense. That's a great question. So in one respect, bacteria, a lot of them are kind of around dormantly. They have like spores, so to speak. And given a plant root, for example, growing into their proximity, they sense that, hey, stuff is here. I'm going to wake up. But also in situations where there's actually water in the soil, they can move pretty good distances. So that's why drought, one of the reasons drought has such an impact is it limits how microbes can traverse through the soil environment and actually get to places they want to get to. But also fungi are really important for this. So mycorrhizal fungi especially, because they grow directly onto roots and they're forming positive associations, can act like highways. And there are actually pretty cool studies and images that have shown bacteria can either navigate through the water film that is on top of these hyphae, so the kind of threads of fungus in these environments, or even grow and kind of move along them through growing on the hyphae to get through the environment. So
any bacteria, including the predator ones?
Not all bacteria. Some the fungus doesn't like, so they'll actively filter out.
So it would probably be somewhat correlating to what ones the plants don't want.
In a way, yes. So a great example of that is actually that they don't actually associate with the plants, but mycorrhizal fungi that grow into the cells, they gave up the otherwise normal fungal lifestyle of digesting things, like degrading stuff in the environment, a long time ago. They can't do that anymore. But they often associate and let bacteria that can form or that can like increase the rate at which phosphorus will leach from organic matter, for example, they'll associate with those, so they can let the bacteria do the hard work of getting all those nutrients out, and then they go, thanks, and they send it to the plant, and the plant gives them sugars and fats in exchange. Yeah, it's a whole lot of relationships going on.
That's fascinating.
At the end of the day, everybody's got to eat. Unfortunately.
Trees have like, you know, like 5 major highway functions, kind of, not really. Basically, it's all going up or it's all going down. But they will try to repair themselves and like reorganize themselves and unblock themselves. And they have to like, maybe the tree got scraped on one side and they'll be like, deadening or pooling or something. And they'll say, oh, well, now we have a highway and that highway shouldn't really be there. And then they'll start sending other things out. So in tree cell studies that I was doing, like chemical paths or environment, soil layer, I think that it was kind of interesting to see different funguses too. It had a lot of comparisons.
Yeah, it's I think it's how do I put it? Like roots especially are like remarkably similar to how fungi move through the soil. That's probably just like one of those life physical constraints, right? There's certain things that promote surface area and navigating complex environments, but especially considering that they were forming relationships with these fungus before they actually had fully developed roots.
Right.
Right, there's a lot of fun creative thoughts to play with there. But yeah, plants as well as fungi, like I said, the roots are kind of like these cities that help change what kind of microbes are growing where in the soil, right? So say, for example, you have a very deep rooted tree, it will allow, because not only are these roots providing nutrients, but they're letting oxygen and other things traverse down into the soil. It can allow microbes that otherwise wouldn't be able to grow much deeper into that soil environment and have changes to the soil chemistry, pH, certain nutrient availability, etc. That if the root wasn't there, it would be quite a different situation. Any other questions?
One last question. How may invasive species, for example, disrupt this balance?
That's a wonderful question. Chaos, that's how. So there's a lot of, like I said, there's a lot of like specificity that can happen with these relationships. So one of the reasons that we actually think legumes evolved this ability, a lot of them are just kind of like really simple, started off as really simple like forage grasses, right? So like alfalfa, for example, or if you ever walked around and you see the little clover that has like little spiny seed pods that grows everywhere, that's a legume. I actually study it in labs sometimes. One of the reasons we think they form this relationship is because it lets them in cases where a lot of plant life has died off or hasn't established yet, where there's not a lot of nitrogen in the soil, relationships haven't kick-started on the processes that let them be available. They have the advantage of going, I don't need that, I can just grow here anyways. And so they can kind of take on and expand out earlier than a lot of other plant species can. So that's an example of that. But also, in the case of like fungal relationships, so the ones that don't look quite like this, that grow on the outside of plant roots that often associate with trees. So it does change in a second. They are much more specific about which kind of species will do that relationship. And a great example of that is eucalyptus trees. Actually have like a very specific relationship requirement. And when they were initially trying to establish eucalyptus trees in California for logging, they wouldn't have it. And they literally had to import soil with that fungus in it to allow the eucalyptus to proliferate here. And then once the fungus got introduced to the soil, eucalyptus went crazy and outcompeted all these other trees. Another example of that is almost every land plant is able to make this association, except a specific group called Brassicaceae. You probably know them broccoli or collard greens or Brussels sprouts or wild mustard, for example. Those plants don't make this relationship. They decided way back when, actually, I don't need that. I don't want to do it. Not my thing. And in fact, because they don't use that relationship anymore, there is actually an advantage for them to actively inhibit it in other plants, which makes them really good invasive species in grassland environments where they can go, hey, you know that network you all like to associate with and do stuff with? Yeah, it's gone. I'm here now. It's my space.
So you'll end up with very low microbial activity,
Or at least
where we have a whole lot of brassicas.
at least these kinds of fungus specifically. Certain other kinds of bacterial that they associate will have like enriched activity. So, you know, nature is all about balance one way or another. So it does kind of balance out, but it's just not what a majority of other plants would need. That's one of the reasons why, if you like look on, I don't know how many emails I've gotten after I've done some of these talks where they're like, how do we get rid of this invasive mustard species? And I'm like, I don't know, man. I haven't done that research yet. You can figure that out yourself. But yeah, mustards can be really, really good at that.
They will inhibit it.
Yes, they can actively inhibit this relationship through certain chemicals they can put into the soil that they exude through they send out from their roots.
So if you wanted to grow broccoli, maybe don't put it in with other types of plants, or is that a concern?
Yeah, I mean, it's definitely like a kind of research thing. But I will say too that in the case of what we grow in agricultural settings, they do operate kind of on a different wavelength than wild stuff. And in garden settings too, depending on what you're kind of how you're growing. So if you're growing like in one spot versus if you're intercropping, or if you're like me, you just are too busy sometimes and just let chaos take reign. Plants will kind of there's certain combinations of like plants you grow next to each other that actually can help each other out in really good ways, in ways that might not be connected to the fungus. They might be connected to certain bacteria species that make certain nutrients available. For example, there's a really cool study with peanuts and one of the other species, but two crop species that they grew together, and one of them recruited a kind of bacteria that made iron much more available for the other one in a way it normally wouldn't be able to. And so they both actually grew much better than they did growing in monoculture isolation. But yeah, long roundabout way.
Should I avoid putting brassicas in my compost in order to?
Oh no, once they're dead and dying, it's different. It's
Is
all
the message out going out through the roots to get rid of them?
yeah, once they're dead, they're not producing all those compounds as much. And then through the process of composting, especially, you're welcoming in all these other bacteria and nematodes and fungi that were here to degrade dead things.
Not a problem. Good.
Yeah, so they just kind of get rid of all those things. The heat too kills a lot of pathogens. And eventually, if your composting goes right, you end up with an environment that's really conducive to the good guys, as we call them, that help to.
Maybe not compost their seed pods.
Well, yeah, actually, it depends on the plant. I'm not as familiar, but yeah, certain plants actually can have toxins to other plants in their seed pods. But I don't
Or
know.
to grow in wherever you throw the compost, the mulch. Yeah. Helpful, thank you.
Have one slide up for the QR code to join our listserv.